Hello, my name is Melanie Frigo, and on behalf of my co-authors, it is my pleasure to present this poster. VIP152, a selective CDK9 inhibitor, induces complete regressions of high-grade B-cell lymphoma models and depletion of short half-lived oncogenic transcripts, Myc and MCL1, with a once-weekly schedule. High-grade B-cell lymphoma, HGBL, is refractory to standard of care chemotherapy and characterized by rearrangements in Myc and BCL2 or BCL6, which drive oncogenic transcription and pro-apoptotic signaling, respectively. VIP152 is a well-tolerated and clinically active CDK9 cyclin-dependent kinase 9 inhibitor, delivering complete metabolic remissions in 2 out of 7 HGBL patients treated with once-weekly 30 milligrams intravenous doses. Herein, we show that an in vitro 4-hour VIP152 pretreatment to mimic the once-weekly dosing delivers durable reductions of RNA polymerase II serine 2 phosphorylation and controls Myc and MCL1 mRNA and protein levels, resulting in cancer cell apoptosis and in vivo tumor regressions. Inhibition of CDK9 results in inhibition of RNA polymerase II phosphorylation at the serine 2 residue, phosphoserine 2. This leads to the reduction of short half-life mRNA levels such as Myc and MCL1. In turn, Myc-dependent oncogenic transcription is downregulated due to the depletion of Myc protein and other oncogenes like MCL1. Potent and durable control of protein levels without continuous dosing delivers an oncogenic shock that results in the induction of cell death and anti-tumor efficacy. VIP152 delivers the greatest extent and duration of phosphoserine 2 inhibition. With a 4-hour in vitro treatment of VIP152, we see 50% inhibition of phosphoserine 2 for 48 hours in the SUDHL4 Myc amplified model on the left, and for 24 hours in the Myc mutant but overexpressed SUDHL10 cell line on the right. In comparison, atuvasiclib treatment leads to a 50% reduction for 16 hours and KB0742 for 12 hours before returning to baseline levels. VIP152 controls RNA polymerase II phosphorylation. We looked at the impact of CDK9 inhibitor treatment on the transcriptome. VIP152 treatment confers the most robust shift in transcriptional activity in Myc-positive lymphoma cell lines, as seen in the 4 volcano plots, 1 for each treatment, and the magnitude of the transcriptional regulation by VIP152, as seen in the 2 left panels, supports an oncogenic shock as a mechanism of action. VIP152 depletes oncogenic short half-life mRNA transcripts Myc and MCL1 using qPCR studies. This initial depletion is followed by some return of Myc and MCL1 mRNA levels. However, the levels of both mRNAs maintains at least a 75% decrease for 48 hours, whereas atuvasiclib or KB0742 treatment does drive an initial decrease, but this is followed by a return to 50% relative levels to baseline. Reflecting the change in mRNA, VIP152 also downregulates Myc and MCL1 protein levels. Unlike the other treatments, 1 micromolar VIP152 delivers sustained Myc and MCL1 protein depletion for 48 hours, delivering an oncogenic shock which creates a temporal window, resulting in pro-apoptotic PARP cleavage as seen in the bottom right panel. In vivo, we see a dose-dependent anti-tumor activity in the SUDHL10 xenograft model, resulting in tumor growth control at 10 milligrams per kilogram and tumor regressions at 15 milligrams per kilogram. After 2 weeks regrowth of the 15 milligram per kilogram group, retreatment is sufficient to suppress outgrowth. The pharmacodynamic effect of VIP152 is also detected in HGBL patient whole blood, which does not contain tumor. The maximal inhibition of all mRNAs occurred within 4 hours of VIP152 treatment. The impact of the presented study is that VIP152 delivers a durable reduction of phosphoserine 2, controls Myc and MCL1 mRNA and protein levels, which results in cancer cell apoptosis and in vivo tumor regressions. The pharmacodynamic effect is demonstrated in the blood of HGBL patients treated with VIP152, and tumor-based studies are planned to confirm the extent and duration of the PD effect. VIP152 is currently being evaluated in HGBL patients and other Myc-driven indications in the clinic. I thank you for your attention.